Hello everyone. I hope you all are following the precautions of COVID and doing well in your lives. Today we will discuss the biology portion chapter 5 of class 9th. In this video we will discuss the solution of the exercise given on page number 6-7 with explanation. The first question is to make a comparison and write down ways in which plant cells are different from animals cell. So, plant cells have cell wall while animal cells don't have a cell wall. Plant cells contain chloroplast while these all things are absent in animal cell. Plant cells do not have any centriole, but in animal cells centrioles are present. When we talk about plant cell, their vacuole is present and that is large and it is present in the center of the cell. When we talk about animal cell, vacuole is present but that is very small. In plant cell, nucleus is present in the side of the plant cell, while in the animal cell, it is present in the center of the animal cell. While you are writing this answer, please mention this diagram also so that everything will be cleared at a glance. The second question is, how is prokaryotic cell different from eukaryotic cell? Prokaryotic cells are generally small in structure, while eukaryotic cells are generally large. Prokaryotic cells has the nuclear region very poorly defined. It means they do not have well defined nucleus and they do not have nuclear membrane, and this structure of poorly defined nuclear region is known as nucleoid. While we talk about eukaryotic, there the nuclear region is well defined and surrounded by a nuclear membrane. In prokaryotic cells, there is a single chromosome, and when we talk about eukaryotic cells, there are more than one chromosomes. In prokaryotic cells, membrane bound cell organelles are absent, while in eukaryotic, membrane bound cell organelles are present. I hope you understood this answer. While writing this answer, please mention this diagram too. You can clearly see the difference here. So, write answer number 2 with this diagram. Moving ahead, question number 3. What would happen if plasma membrane ruptures or break down? I hope that you all know that what does a plasma membrane do. If not, just see that there are the function of plasma membrane. These are not the part of answer, but I am just explaining it. Plasma membrane provide the cell a selectively permeable barrier or boundary. उसे चारों तरफ से ऐसे कवर किया रहता है. It separates the interior of the cell from the external environment and it provides a layer of protection too. It regulates. It means they also work as a traffic police. They regulate that what materials or molecules can enter or exit the cell. It provides the point of attachment to the other cell, and this is very important for the cell to cell communication. It means you all must understood that this is present at the outermost structure before cell wall, and it provides a protection to the whole cell. So you can imagine what will happen that if it gets ruptured like this. You can clearly see this in diagram that the membrane get ruptured and all the cytoplasm get disturbed here. So what will happen? You will write this answer. If plasma membrane ruptures or break down, then molecules of some substances will freely move in and out of the cell because plasma membrane acts as a mechanical barrier. So exchange of material from its surrounding won't take place, and consequently the cell will would die due to the disappearance of protoplasmic material. It means प्लाज्मा मेम्ब्रेन अगर फ्रैक्चर होती है, ब्रेक डाउन होती है, तो कंसिक्वेंटली क्या होगा कि धीरे धीरे उसके जो मटेरियल्स हैं वो आउट होने लग जाएंगे एंड द सेल वुड डाई. Moving ahead, the fourth question: what would happen to the life of a cell if there was no Golgi apparatus? So I think you all know that how does a Golgi apparatus look. This question, when you are going to answer this, please mention this diagram also. Here the cisternae, cis-phase, lumen, transphase and vesicles are present. So Golgi apparatus, basically a packaging material for the cell. What does it do? It stores the substances, it packages the substances and also manufacture the substance. Without the Golgi apparatus, the cells will be disapplicable for packing and dispatching materials that were produced by the cell. So it is also involved in the formation of cells. Therefore, in its absence, cells will be also not get produced. So all these works will be get affected. So you can write this answer. Moving ahead, the question number 5. Which organelle is known as powerhouse of the cell and why? I think you all must know that mitochondria is known as the powerhouse of the cell. Why? If you don't know, then I just give you some revision points. That this is a double membrane structure. It have its own DNA and it's very numerous in high required cells, muscles, where the continuous supply of oxygen is required. Also, what are their function? They are the powerhouse of cell. It transfers energy to the ATP to be used for the life functions. It is found in eukaryotic as well as in the prokaryotic cells. Also, when you write the answer, please mention this diagram too, this diagram of mitochondria structure. Moving ahead, the answer will be mitochondria are known as the powerhouse of the cell. It is because it releases the energy required for different activities of life and it releases energy in the form of ATP. ATP is also known as energy currency of the cell. So the answer will be mitochondria and you will write this answer. Moving ahead, question number 6. Where do the lipids and proteins constituting the cell membrane get synthesized? It means you just want to ask that which organelle is responsible for the cell membrane synthesis, the synthesis of lipid and proteins. So you will answer the lipid and proteins are synthesized in ER, which is the short form of endoplasmic reticulum. What is endoplasmic reticulum? Just for the revision purpose, I am going to say a few points. Like, it make and packages protein and lipids. It works as an assembly line. It means it works in the packaging of everything. It is found in eukaryotic organisms especially. It forms an interconnected network among the cells. This is the largest organelles in eukaryotic cell. इस बात को ध्यान रखिएगा कि जो largest organelle है that is your endoplasmic reticulum. It provides a separate chemical environment which allow the correct protein folding. It is of two types, rough endoplasmic reticulum and a smooth endoplasmic reticulum. Please mention this diagram where you can see that the places where ribosomes are present are known as rough endoplasmic reticulum and wherever these are not present these are smooth endoplasmic reticulum. इस फिगर को प्लीज मेंशन कीजिएगा अपने आंसर में. दिस इज वेरी इंपॉर्टेंट. मूविंग अहेड, द क्वेश्चन नंबर 7. हाउ डज एन अमीबा obtain its food? Through the process of endocytosis. You must have studied this in earlier classes also. This is the structure of amoeba through the microscope, and it takes its food through endocytosis, where what happened, with the help of pseudopodia, it traps the food vacuole, and then secretion of enzymes occur, and this whole food get dissolved. So we can write, through the process of endocytosis, an amoeba obtain its food, and its cell membrane is flexible, so the food particles get engulfed matlab that get surrounded by pseudopodia, and there the food vacuoles start grinding, and it will be completely dissolved with the help of enzymes. Okay, so mention this diagram too. Moving ahead, the question number 8, what is osmosis? Osmosis is the process of movement of a water molecule from a region of higher concentration to the lower concentration to the semi permeable membrane. This is very important, that osmosis will always takes place in the presence of semi permeable membrane. How does it happen? So when you are going to write this answer, please mention this diagram too. You all can see that here the concentration is low, and the another side there is the high concentration, and between both rays there is a membrane which is known as semi permeable membrane. The solvent molecules start moving from the lower to higher solute concentration. इस चीज़ का ध्यान रखिएगा, यहाँ पे मूवमेंट जो हो रहा है वो सॉल्वेंट्स का हो रहा है. ओके, इस फिगर को जरूर ध्यान से मेंशन कर लीजिएगा अपने आंसर में. एक मैं आप लोगों को एक्स्ट्रा बता देती हूँ कि व्हाट इज़ द डिफरेंस बिटवीन डिफ्यूजन एंड ऑस्मोसिस, बिकॉज इट्स क्वाइट कन्फ्यूजिंग. इसलिए इन डिफ्यूजन, मूवमेंट ऑफ मॉलिक्यूल्स फ्रॉम द हाई कंसंट्रेशन टू द लो कंसंट्रेशन. ठीक है, ये तो आपका सही है. लेकिन यहाँ पे सॉल्यूट और सॉल्वेंट दोनों ही मूव होते हैं, नॉट ओनली सॉल्वेंट. बट व्हेन वी टॉक अबाउट ऑस्मोसिस, हियर इज द मूवमेंट ऑफ सॉल्वेंट ओनली. I think you must know the difference between solute and solvent. Here only the movement of solvent occur from a region of high to low solvent concentration. Only solvent molecules move. In the 9th question, there is an osmosis experiment. आप लोग में से कुछ लोग अगर चाहें तो इसे अपने एक्सपेरिमेंट को अपने घर में भी कर सकते हैं, एंड इट विल बी अ गुड एक्टिविटी. आई विश कि आप में से कोई भी इसे एक्सपेरिमेंट को करें और एक्सप्लेन करते हुए एक वीडियो शूट करके ऑफ 1 or 2 मिनट क्लास ग्रुप में शेयर करें. सो हियर व्हाट इज द क्वेश्चन? You have to take 4 peeled potato halves and scoop each one out to make the potato cups. One of these potato cups should be made from a boiled potato, and put each potato cup in a trough containing water. So how you have to do? You can understand this with this diagram. आपको 4 पोटैटो के हाफ्स लेने, मतलब ऐसे एक पोटैटो के बीच से काटे तो 2 हाफ्स हुए ना, उस तरह से ऐसे 4 पार्ट लेने हैं. एंड देन उसे बीच में से, बीच का जो उसका पार्ट है, जैसे हम लोग जब आइसक्रीम लेने जाते तो ऐसे एक स्कूप निकाल के देते हैं ना, वैसे ही एक बस आपको स्कूप इसके ऊपर में से हटा देना है. इनमें से जो 3 पार्ट रहेगा वो तो आपके रॉ पोटैटो रहेंगे, लेकिन एक जो रहेगा वो आपका बॉइल्ड पोटैटो रहेगा. ठीक है, तो आपने कैसे बनाना है? 4 आपने हाफ्स करने हैं, बट 3 ऑफ विल बी द रॉ पोटैटो, और जो 4th वन रहेगा आप एक पोटैटो को बॉयल करके उसको हाफ करेंगे, उस चीज़ को आप यूज करेंगे यहाँ पे. देन आपको क्या करना है? पुट एक, यहाँ पे हमने इसकी नेमिंग कर दी कि 1 हाफ इज ए, अनदर बी, अनदर सी, अनदर डी, लाइक दिस. एक को हमें एम्प्टी रखना है, वी विल नॉट कीप एनीथिंग इन इट. सेकेंड में हम 1 टीस्पून शुगर डालेंगे. यू कैन यूज़ दैट ग्राइंडेड शुगर ऑल्सो, उसको आप पीसी हुई जो शुगर रहती है आप उसको कप बी में डालेंगे. देन कप सी में यू विल पुट 1 टीस्पून ऑफ सॉल्ट. कप डी में जो कि बॉइल्ड पोटैटो था हमारा, वी विल पुट 1 टीस्पून ऑफ शुगर. अब हमें इसे 2 घंटे के लिए रख देना है. देन जब हम इसे ऑब्जर्व करते हैं तो व्हाट वी फाइंड दैट वाटर गैदर्स इन द होलोड पोर्शन ऑफ बी एंड सी. बी और सी का जो ये होलोड पोर्शन था नीचे का, यहाँ पे वाटर गैदर हो गया है. वाई इज पोटैटो ए नेसेसरी फॉर दिस एक्सपेरिमेंट. द क्वेश्चन इज कि जब हम पोटैटो ए में कुछ कर ही नहीं रहे हैं तो क्या जरूरत है कि हम उसे एक्सपेरिमेंट का पार्ट बनाएं. एंड थर्ड क्वेश्चन इज एक्सप्लेन वाई वाटर डज नॉट गैदर इन द होलोड आउट पोर्शन ऑफ ए एंड डी. और ए और डी के पोर्शन में वाटर क्यों नहीं गैदर हुआ, जो कि बी और सी में हो चुका था, तो ए और डी में क्यों नहीं हुआ. दिस इज द क्वेश्चन. प्लीज मेंशन दिस डायग्राम एनी ऑफ टू. यहाँ पे ये 2 मैंने डायग्राम बनाया, जो कलर वाले और ब्लैक एंड वाइट, दोनों में से कोई भी जो भी आपको अच्छा लगे वो आप डायग्राम अपनी कॉपी में बना सकते हैं. दिस विल हेल्प यू टू अंडरस्टैंड दिस क्वेश्चन एंड ऑल्सो दिस विल हेल्प द एग्जामिनर टू कि आपको क्वेश्चन अच्छे समझ में आ गया एंड यू मे गेट एक्स्ट्रा मार्क्स टू. Moving ahead, the solution. What is the reason behind all these things? In the first question, the question was that explain why water gather in the hollowed portion of B and C. So the solution is water accumulated in the hollowed portion of B and C because of difference of water concentration. आपने अभी भी osmosis का process देखा है. You have seen that solvent particles moves from the लो कंसनट्रेशन टू हायर. वहाँ पे वाटर हमारा सॉल्वेंट था, तो जब यहाँ पे हमने शुगर ऐड कर दिया तो ऑब्वियसली वहाँ का कंसनट्रेशन हाई हो गया, तो वाटर मूव करने लग गया. दैट्स वाई इन द पोटैटो नंबर बी एंड सी द वाटर स्टार्टेड गैदरिंग. वाई इज पोटैटो ए इज एसेंशियल? पोटैटो ए इज एसेंशियल इन दिस एक्सपेरिमेंट as it is significant to compare. This is important. Why? To compare that how is the process of osmosis occur in other potatoes but not in this. What is the significance of potato number? The third question is that explain why water does not gather in the hollowed out portion of A and D. क्वेश्चन ये है कि जो A और D के hollowed portions है, यहाँ पे वाटर क्यों नहीं गैदर हुआ. So the answer is cup A does not show any change because it required the concentration of higher that side. तो वहाँ पे कोई ऐसा तो कंसंट्रेशन हाई करने के लिए ना तो वहाँ पे सॉल्ट ऐड किया गया था, ना शुगर नहीं, और कोई सॉल्यूट कुछ भी वहाँ पे ऐड नहीं किया गया था. दैट्स वाई वो वहाँ पे बिल्कुल भी वाटर गैदर नहीं हुआ. एंड व्हेन वी टॉक अबाउट डी, बिकॉज दैट गेट बॉइल्ड. आपको याद होगा कि एक जो हमने जो डी पोटैटो रखा था दैट इज बॉइल्ड. When we boiled that, जो उसकी सेल्स थी वो डेड हो चुकी थी, इसलिए यहाँ पे कोई भी सेमी परमेबल मेम्ब्रेन नहीं बची जो कि वाटर फ्लो को रेगुलेट करे, और कंटिन्यूसली यहाँ पे ऑस्मोसिस भी नहीं हुआ. सो यू कैन डू दिस एक्सपेरिमेंट एट योर होम and you can shoot a video of 1 or 2 minutes where you have to explain about the osmosis process and the presentation of osmosis. Okay, moving ahead, the 10th and the last question. Which type of cell division is required for growth and repair of body and which type is involved in formation of gametes? There are 2 ways in which a cell divides. 2 तरीके से सेल डिवाइड हो सकती है, एक माइटोसिस, दूसरी मियोसिस. माइटोसिस क्या होता है? It is the type of cell division where the growth and repair of body occurs, whereas meiosis is a type of a cell division which results in the formation of gametes. It means mitosis में क्या होता है कि बेसिकली जो सेल डिवीजन होता है उससे 2 डॉटर सेल्स फॉर्म्स होती हैं, और यूजुअली ये आपकी जो रिप्रोडक्टिव सेल्स रहती है उसमें नहीं मिलता है. ये यूजुअली बोथ आपकी बॉडी की ग्रोथ और रिपेयरमेंट के लिए माइटोसिस रिक्वायर्ड है. बट meiosis where 4 daughter cells are formed, this occurs in the reproductive cells only. इफ द क्वेश्चन दिस इज फॉर एक्स्ट्रा नॉलेज कि अगर कोई आपसे पूछे कि व्हाट इज द डिफरेंस बिटवीन माइटोसिस एंड मियोसिस, सो यू कैन आंसर दैट माइटोसिस occur in the place in the somatic cells, जो somatic cells रहती है उसी में माइटोसिस मिलेगा. And meiosis it will occur in the gametic cells or reproductive cells. In the mitosis 2 daughter cells are formed and meiosis 4 daughter cells are formed. In mitosis number of chromosomes remain diploid in daughter cells and number of chromosomes become haploid in daughter cells. In the mitosis is necessary for the growth and repairment of body. Meiosis is necessary for the sexual reproduction. Mitosis, in the process of mitosis, crossing over does not take place, and in meiosis crossing over takes place. This crossing over is a part of the higher classes. क्रॉसिंग ओवर के होने की वजह से ही यहाँ पे नंबर ऑफ क्रोमोसोम्स जो है दैट रिमेन डैप्लॉइड, क्योंकि यहाँ पे कोई क्रॉसिंग ओवर नहीं हुई है, बट हियर क्रॉसिंग ओवर अकर्स, दैट्स व्हाई द क्रोमोसोम्स बिकम हैप्लॉइड. सो बेसिक चीज़ ये है कि माइटोसिस टेक्स प्लेस इन द सोमैटिक सेल्स एंड इट इज़ रिक्वायर्ड फॉर ग्रोथ एंड रिपेयरमेंट ऑफ आवर बॉडी, एंड meiosis takes place in the gametic cells or reproductive cells. Here 4 daughter cells are formed. Okay, now moving ahead, I think all questions are completed. Now when you are going to answer this question number 10, please mention this diagram too, that how does 2 daughter cells are formed through mitosis and through meiosis 4 daughter cells are formed. Please mention this diagram too. So I hope that you have understood all the questions asked in the exercise. If any doubt remained, please clear your all doubts by asking in next class. So thank you so much for watching. Like, share and subscribe this video. Take care a lot. Stay home, stay active and stay safe. Thank you.